Hello everyone, I am Marco Martin.
And I'm Miriam Corcho.
And welcome to Bradley Forensics.
Today we will discuss a crucial process in forensic investigations.
That's right, we will be talking about autopsies.
What is an autopsy and why do we need them?
Well, autopsies are medical examinations that are performed in a dead body in order to determine the cause of death.
Autopsies might be required for special cases such as a homicide or even simpler ones such as a heart attack. This is because an autopsy concludes the cause of death of the person.
That's right, autopsies go above the forensic field. But for those who pertain to us, they're related when a body is found and the cause of death is suspected or completely unknown. I'd like to start by saying that autopsies and dissections are similar, yet not the same thing.
Although they both pertain to a close inspection of the external and internal factors of the body, autopsies are used to determine the cause of death.
Yeah, you might be more familiar with dissections, as they're commonly used in biology classes to study the internal organs of animals like frogs.
However, today we will be doing an autopsy using human standards on a pig.
Wait. What do you mean following human standards? How are you going to follow human standard if it's not a human?
That's an excellent question. That's because when it comes to body autonomy, humans and pigs are very similar, as we have organs in similar positions and we share organ functions with them.
Oh, that's right. Whereas if you were to use a cow, you will be thrown off because their stomach is divided into 4 compartments.
But don't worry, that's not what we will be doing today.
We also understand that autopsies and dissections are not available to everyone and should only be performed under certain circumstances.
This is why we advise students and teachers to find another alternative to this activity.
Some of these alternatives include virtual dissections and virtual autopsies, which are available online, and for some of those, we will be providing the link in the description of the video. It is within the laws of our school district to allow students to perform dissections in the classroom.
This is as long as the activity is for the purpose of the education of the students and everyone participating will consent to it.
Consent involves students, teachers, and parents revising, signing, and understanding the guidelines for the use of animals in secondary science activities.
We have already provided the students with a copy of this document for them to revise with their parents and sign appendices 3, 4, 5, 6, and 7.
In addition, we also ask students to sign a media release form, which they and their parents have to sign. This guarantees that we can share our content here on YouTube with you.
Still, if you are sensitive to any content that involves dissecting animals, animal death, bodily fluids, or any grotesque content in general, we advise that you do not watch this video.
But we will catch you on the next one for sure. Before we jump into it, let me show you the materials that we will need for this experiment. We're going to require a scalpel, a pair of tweezers, surgical scissors. These are specifically surgical scissors because the tip of them is rounded. We're also going to need pins to secure the body to the mat. And talking about mat, we need a tray with a silicone mat where we will place the body. In addition to this, we will need little containers where we will place each one of the organs. And a scale to weigh each one of the organs independently.
And most importantly, all the personal protective equipment that we will need, which will be a face mask, a pair of eye protection, gloves, and an apron.
And wait, don't forget Clorox wipes, because after that we were gonna have to clean everything.
Now that we got our materials, let's jump into it.
And here we are in full PPE.
Personal protective equipment is used to protect the pig from any contamination that our bodies will bring, as well as protect our own bodies from any contamination that the pig will bring. Let's begin our autopsy by bringing the pig into the mat.
Well, as you can see here, the pig is completely unclothed, and this is what we get from the crime scene. Because if you might remember from our crime scene video, the body, when it's found at the crime scene, it is completely unclothed there.
This means that what arrived at the medical examiner's office is an unclothed pig that has been partially cleaned.
That is correct, because crime scene technicians need to unclothe the body in order to record any potential evidence that they might find on the surface of the body.
They also photograph, note, and collect any evidence or any other potential evidence like mud or paint that is found on the body, especially under the fingernails.
This is for them to ensure that any evidence that they find on the body is preserved. Because as we receive the body will be completely clean and is going to go through different processes that might damage any of the evidence that is found on the surface of the body. The medical examiner's room does not get the cloth from the body. That is sent to the trace evidence section of the crime lab and potentially to the DNA section in case that they suspect of any presence of DNA in the cloth. That's why we only get the body. We do not have to handle any cloth, any article that item that comes with the body. We just get the body completely unclothed.
Before we jump into our first step, we will turn on the voice dictator.
In this case, we really don't need a voice dictator because we will be recording the entire process for you guys. And we will get not only a voice dictator, but also the image of the entire process.
Our first step is cleaning the body.
Cleaning the body is detrimental as we will later be passing the body through different examinations. And we want to make sure that the body is as sterile as possible. So the results that we gather from those examinations are reliable.
Since our body is a pig obtained from a reputable source that sterilizes these pigs and preserves them in a vacuum sealed bags, we do not need to clean the body.
Does it even get any cleaner than this? I mean...
Okay, let's move on to step 2.
Our next step is doing x-rays of the body.
And who told you we have an x-ray machine?
I know we don't have an x-ray machine, but this will be what you will normally do. X-rays do not only let you know if there are any bone fracture, but also they can help you determine if those bone fractures penetrated any crucial organ, along with determining if there are any foreign objects inside of the body.
Foreign objects don't only include bullets or nails, it also could include hip replacements, screws, metallic plates, or anything that has to do with medical interventions.
And yeah, let me tell you, these medical interventions, if you find any in anybody, you just hit the jackpot because they're detrimental to individualizing the suspect.
Moving on, we will do an external examination of the body, photographing, noting, and considering every relevant physical characteristics that the body has.
We will note their height, race, color, skin color, hair texture, the weight, and all of these characteristics we'll be providing them on screen for you right now as we had previously noted them, and we'll put them on screen for you to see it.
These are generic characteristics that will help conduct an age approximation of the body that will also help us facilitate their identification.
However, if the body will come with any scars, tattoos, piercings, any scar from a medical intervention, you just won the lottery. Because all this scar will help you individualize the suspect, making it easier to identify them in the case that you just found a body and you don't know who this person is.
Other important factors that will help in the examination will be any wounds or bruises present in the body, since this will help locate the cause of death.
As you can see here, and as we had previously noted in our first visual examination of the body, they do not have any physical and any visible wounds, tattoos, piercing on any marking that would help us identify them. And let me tell you something else. The x-rays already came back and the results show that there are no bone fracture and there is none of any other external item inside of the body. So this leaves us with a very good mystery of what happened to this person right here.
So let's get into the actual autopsy because we have a lot of work to do.
And this is where the fun begins.
To dive into the internal organs of the person, we will start by making a Y-shaped cut in the thoracic cavity, starting with a diagonal cut from the clavicle all the way down to the center to the chest on both sides of the body. Both of these cuts must be in the center of the chest. from where we will continue to cut all the way down to the pelvic region, except that at the umbilical region we will cut in a circular shape to prevent from destroying the belly button and the bladder, which is positioned right under.
This first cut has to be very superficial. It only intends to perforate some of the skin and some of the abdominal fat. This ensures that we do not go too deep and break into some of the internal organs in this first superficial cut.
After the initial cut is completed, we can go and perform a deeper incision that perforates the abdominal muscles and lets us enter the thoracic cavity. This cut can be made with a scalpel or a pair of surgical scissors. Once the incision is completed, we can pull the skin and muscles of the abdominal region to expose the organs.
Perforate. I am breaking into the abdominal muscles. They are a little bit harder than the actual skin. So you can see here, I'm struggling a little bit. All right, there. That one cut all the way down. There we go. All right. In the case that you struggle to still open the abdominal cavity, you can continue in making other incision right here. This incision will turn into an H shape.
We must remember that this is a person we would be performing on, and we want to cause the less damage as possible in order to return them to their relatives in the best conditions possible. So only do this if it's completely necessary.
In this case, we use specific scientific dissection techniques for a pig's anatomy because a pig's structure is different from the human, and we gotta compensate, and we also need to get a better view of the internal organs. As if we only did the wide cut shape, you still get a good view of the organ, but it's as if it is less invasive. You're going to get less visibility of while the organs are inside. This also helps us get a better angle with our camera.
Now we will be following the Rocky-Tanki method to remove all the internal organs, starting from the thoracic cavity and working our way down. It's important to place organs in a separate container once they are removed from the body. That way they can be examined separately. Starting from here, let's notice the main organs that we will remove. Those are the lungs, the heart, the liver, the stomach,
Over here on this side,
the small and large intestines.
the small intestines are over here, and the large intestine is hiding a little bit on the side over here.
And once these are removed, they will reveal the kidneys, which are hiding in the back of all of these organs. To remove the heart, we will locate the pericardial sac, which is a membrane that protects the heart. We will cut this membrane to expose the heart and then locate the ventricles and vessels.
So you can see here, the membrane comes out with a little cut. You don't have to do much. I'm gonna hold it with my tweezer so I can pull it out once I cut it.
We will cut these connections that the heart has to the rest of the body and remove it.
I speed it up right there because as I was cutting the ventricles, and you gotta remember that there are also still going to be some ligaments that are holding the heart in place. You also gotta account for those ligaments, not only the ventricles. So I had to went and carefully cut the ligaments that were holding the heart. As you can see over here, let me use my scalpel to help you notice. Those ligaments are holding the heart in place. And I also had to account to take out those. Now that I finally cut all of that, I'm just missing something over here, and there you have it. That is the heart. We're going to place it in its individual container, and we will continue into our next step.
The lung can be easily lifted and cut the trachea to remove them. The procedure that we will follow for bilateral organs is different from other organs. Bilateral organs are organs that mirror each other, meaning that you have two of these, one in the left and another one in the right of your body. When they are removed from the body, they must be placed in different containers, differentiating the left and right lungs.
What I did right there was cut the trachea, and now I'm going to extend all the way down here. I am going to start with the right lung, right? I will be cutting all those membranes that held the lung together to the walls of the thoracic cavity. There you go. Got it free from over there. You can see that the lungs stand all the way to the back, and here is joining. I want you guys to be able to see this better. Look, you can see right here how the membrane breaks. That membrane is holding the lung together to the walls of the thoracic cavity. And down here, there is another one. I can feel it. Look, right there. This one, I'm gonna actually use this copper to take it out. And there you have it. This is the right lung. I'm gonna put it in this container. I'm gonna give it to them so they label it. That way we don't get lost of which one is the right lung and which one is the left lung. Right, let's continue now to the left lung. I am going to take this membrane that you can find over here. This is the diaphragm, right here. This is diaphragm, this is diaphragm. Right, the diaphragm is holding the lungs as they are right under the lung. Okay, over here, I'm going to cut the same membrane that I show you on the other side. I just did, so let me see if I can take a good try and get it out. There you have it, the other lung. I'm gonna give it to them again so they can label it. That way we don't get lost in which one pertains to one side and which one pertains to the other. As you can see over here, the thoracic cavity is completely clean. So what I'm going to do before we continue is to get rid of the diaphragm, so it doesn't bother us as we work our way down.
To remove the liver, start by severing the ligaments attaching the liver to the diaphragm and the anterior abdominal wall with careful delivery cuts. Also cut any blood vessel that are attached to it.
As I had gotten rid of most of the diaphragm before, you do not see it as well, but you can see right there that this part of the diaphragm and the esophagus was being held to our liver, so I just got rid of that. I can lift here the liver. You can see there is a ligament that is holding the liver together to the stomach. I'll make an incision right there. It's gonna let me continue my way down. Gently cut right there. I don't want to cut into the liver, nor I want to cut into the stomach. I want to get them as preserved as possible right there. So you can see our liver extends all the way down here, and I don't know if you can see, maybe if I move the small intestines, it is being held right there. So what I'll do, I am going to lift it a little bit with the tweezers so I can easily see where the ligament is located. And I put my hand over here, that way it's better for you guys to see. And look, an easy cut right there, and boom, got the liver out. This is it. Very large liver. I'm gonna put it in its separate container, hand it over there, and let's continue.
As we continue down, we would remove the stomach now. Gently search for the connection of the stomach to the small intestines and cut it. Later, ensure that there are not any ligaments connecting the stomach to the walls of the abdominal cavity. This way, the stomach would easily come out.
All right, so as Miriam said, this is the point where the stomach, as you can see, which is this mass that you have over here, connects to the small intestines. The small intestines continue over here, blah, blah, blah. And I am just going to make an incision over there. There is a lot of ligaments that are pressing around the stomach. And you have to go very carefully removing all those ligaments. The reason why you have to be extra careful with the stomach is because the stomach works as a sack, meaning that it's going to be very full of the food that you ingest inside. Okay, right there, I'm making another incision. And if you get to cut it by any reason, it is going to release all the content that it has inside. Right there, I am just missing to make another incision here. And here. Here I can see where the esophagus is getting into the stomach, so I know that that is the point where I have to make a harder incision because the esophagus is a little bit thicker than all the other ligaments that are connecting the stomach. Stomach, as you can see it over here, a little mass, and I'll hold into them.
Both the large and small intestines are intertwined by ligaments that hold them together and attach to the walls of the abdominal cavity. You only need to cut the ligaments that attach it to the walls of the abdominal cavity. Also cut the connection of the large intestine with the rectum, the way both intestines should come out easily. These are the only organs that receive a different treatment, as they are a combination of organs. They are only separated and further dissected to find clues about the diet of the body or any severe stomach condition.
As she had mentioned, you can see over here, this is the point where the large intestines continue to the rectum. This portion, I'll try to take it out as much as I can without breaking into the large intestine. And as she had also mentioned, this is the point where the stomach connected to the small intestine, which we had already cut to take out the stomach. And if you continue, there is a little mass right here. Let me show you with this. Here. There's a lot of ligament present right there because that is the ligament that is holding the large intestine together with the small intestine. As you can see over here, this is part of the large intestine and it's all intertwined in this mass of small and large intestines. So what I'll do right now is that I'll start from the top all the way down, trying to be very careful and remove this portion of large intestine that we have right here. Look, I'm gonna use it from over here so you can see the kind of cut that I'm doing. I am going with a very superficial cut. It is just a matter of breaking those ligaments that hold the large intestine all the way down to the rectum, and it will peel off. Okay, I left it a little bit. I want to get as close as I can to the rectum. And once I cannot get any closer, I just break. And there you have it. This is the mass that pertains to the small and large intestines. Separating the small from the large intestine might be required in some cases. However, it is extremely difficult. As you can see, there is a lot of ligaments that are holding the small intestines together. Kind of looks like a brain if you think about it. That way, it is very hard for us to go back and follow all those little details to take out the large and small intestine in order to separate them. And that is the reason why we would not be doing that, but in a regular autopsy, it is required to complete those steps. However, for us, it will be, number one, too difficult, and we do not count with appropriate training to do so. And number two, it will be time consuming. So I will place them on their little jar, or together, and I'll hand them over so they can classify them.
Now that the stomach and both of the intestines are removed, they reveal the kidneys. They are wrapped around these tissues that secure them to the dorsal part of the pig, which in another word is the back of the pig. After removing these ligaments, you can easily cut the connections of both kidneys to the bladder and carefully remove them. Kidneys are also bilateral organs, as well as lungs, meaning that they will be placed in separate containers, differentiating the left and right kidneys.
I just very, very carefully go over there with the scalpel, trying to make an incision right there. You see that incision? And this tissue that is wrapping the kidney is going to come out easily. I'm going to show you right now. I'm going to finish the cut all the way down here. And look at this. I lift it, and there you have it, your kidney. So, since I want to make sure that I don't damage the kidney, I want to get rid of, number one, this tissue that is bothering here on the way. All right. And number two, I want to search a little bit for the connection of the kidney to the rest of the urinary system. That is the connection that I want to cut into. For example, like this right there, that's ligament. That is not the connection that we're looking for. We remove that. And there you have it. This is the kidney. This is the connection of the kidney to the rest of the urinary system. There's this little tube. There's a lot of ligament over there. Maybe that's why you don't see it the same way that you will see it in a biology book. But let me tell you, this is the real world. This is not a biology book. That's why it doesn't look pretty. It's not color coded, unfortunately. So I'm gonna go ahead, cut that connection. There's still some ligament. I swear I cannot make this up. Ligaments are everywhere. But well, they play a crucial role, which is holding our organs in place. That way, if you walk around, you jump, you do this, you do that, your organs are not just going to get smashed among each other. There you have it. That's the kidney. And this is what connects your kidneys to the rest of the body. Look, it's a little tube. So that is the left kidney. I'm gonna place it over here. Now I want to continue, and I'm going to follow the same procedure to take out the other kidney. I want to go in very carefully because remember, our main goal is to study the organs once we take them out of the body. That's the reason why we take them out of the body in the initial. And if you damage the organs where you're trying to take them out of the body, it will be harder to examine them, as you will have to account for the damage that you brought to those organs. That is the reason why medical examiners have a very important job, which is being extremely talented and extremely knowful of anatomy in order to be able to take off all these organs without damaging them. And let me tell you, from a person who's doing it right now, it is extremely difficult. It's extremely difficult, mainly because I don't see anything. As I told you, this is not a biology book. You cannot see it color-coded. You don't know what you're doing. You have to stop. You have to clean. You have to stop again. You have to clean again. You have to take out ligament. You have to move. You gotta push around. It is very difficult. But that's the reason why these people get paid for what they do, and they get paid really good, let me tell you. That's an excellent career that you can take, and they do get paid really good. So right now, over here, I did the same thing. I found where the kidney is connected to the rest of the body, and I am going to make an incision. And there you have it. I was able to take this one pretty good too. Wow, I'm proud of myself. Look over there. This is the tube that connects the kidney to the rest of the body. Look. Another kidney in perfect conditions. I'm gonna hand it over. And let's move on to the next step. Now, let's cut into the brain. Start from one ear. We're going to go around all the way around the head up until we get to the other ear. And I'm gonna show you something pretty cool. Once you have finished this initial cut, your skin peels back like nothing, revealing the cranius. And this is going to be the hard part because you have to break into the cranium and in order to take it out completely and access the brain. As you can see right here, we got perfect access to the brain. You can see that the two division of the brain. Beautiful. Now we have to take it out. This part you gotta be more careful about it, especially because the prefrontal part of the brain of the pig is going to be very smooth. That part is going to be like a little bit more jelly-like, especially also the lower section of the brain is also going to be smoother. On the other hand, the occipital part is going to be harder. You don't have to worry as much about it. As you can see right there, I was able to completely remove the brain of the pig. It came in perfect conditions. I cannot say how proud of myself I am right now. But yeah, this is definitely a procedure that is a little bit more hard compared to the other organs, especially because your brain, it is more soft in comparison to the other organs. So right there, we have it. We're just gonna put it to the side, classify it, and follow the same procedure. Before we call it a day, let's collect DNA samples from this person.
This process will vary depending on the stage of death that the person is in. So let us explain those stages to you.
There are 6 main stages of death that a person goes through after death. And depending on which stage of death the person is already found, it will be the technique that we will use to retrieve DNA from them. For instance, if the body is found in the first or even the second stage, now known as polymortis or algomortis, it is more convenient to retrieve a blood sample.
However, if the body progresses into the third and fourth stage, which is the livor mortis or the rigor mortis, it's harder to work with the blood as it starts to coagulate. So it's better to get both a saliva sample from a cheek swab and a blood sample.
Once the body reaches a stage of decomposition, there will be less blood present in the body, as liquids are among the first to leave the body. In these scenarios, it will be more convenient to retrieve a piece of flesh. However, it may be the case that due to a high level of decomposition, all the flesh that you can encounter has a high level of contamination. In those cases, it will be more convenient to retrieve both a sample of flesh and a sample of saliva from the mouth.
Lastly, if the body has reached an advanced decomposition or the
skeletonization stage,
it's better to just retrieve a sample from the bones or the teeth. The reason we removed each organ from the body is to allow for a better visual inspection outside of the body.
You might notice that when the organs are inside of the body, they're very difficult to see because they're piled up against each other.
However, now that we have them outside and have cleaned off all of the bodily fluids, we can examine them much more clearly.
Let's go do that. The first organ that we will be inspecting is the heart. Let's start by saying that this pig had quite a big heart. At the top of the heart is where both the aorta and the vena cava are located. Moving towards the lower section of the heart, where this pointy end is, we can find both of the ventricles, the right ventricle and the left ventricle.
As we can see in this case, there is no visible trauma to the heart. Additionally, the weight of this organ suggests that the pig had a healthy heart condition at the time of death. Further dissection of the heart might be required to rule out any cases of heart attack, coronary artery disease, or other heart-related conditions.
The second organ that we will be inspecting is the lungs. As we have mentioned several times, the lungs are bilateral organs, so we must inspect them separately. Let me start by showing you the left lung. Right here we have it, and then I'm gonna bring into the table the right lung. Perfect. So I want to start by saying that there is a big misconception. Many believe that lungs are one single rounded layer. In reality, they're composed of different lobes stacked one on top of each other, as you can see over here. You can see over here, this is one lobe. This is the upper lobe. Then you're gonna have the middle lobe, and then you're going to have the lower lobe. They're all connected to the trachea, which in this case, you cannot see the trachea because I cut it to remove the lungs. But in the pig, when we were doing the dissection, we noticed that the trachea came from connected these two lungs at these two endpoints that you can see over here, and then it extended all the way to the mouth to the nose of the pig.
As you can notice in this case, there is no significant pigmentation or staining anywhere in the lungs, ruling out potential signs of lung cancer or other diseases. However, further dissection of the lungs is still necessary to rule out cases of suffocation or drowning.
And you want to know a cool fact? Forensic scientists can determine whether the person suffocated or drowned not only by looking at the organs, meaning not only by looking at the lungs, but also by examining other nearby context clues.
That's because in the case of traumatic suffocation, the petechiae, tiny hemorrhages or super small blood vessels, are broken due to high intravascular pressure. On the other hand, in drowning cases, the analysis becomes a bit more complex. It's not just about finding water in the lungs. You also have to consider whether other trauma might have incapacitated the individual and allow water to enter the body post-mortem.
That's correct. But it doesn't seem like it's going to be the case over here. These lungs are healthy. They weighted in a proper way. This pig seemed to have a healthy lung condition, so we can definitely discard any lung-related disease. The next organ we will be inspecting is the liver. The liver, as we can see it over here, is very prominent, the liver. It is divided into left, well, in this case, that would be the left of the body, and the right lobe. During our inspection, we look for enlargement and discoloration in any region, as these findings could indicate conditions such as hepatitis or cancer. Another important aspect to consider is whether there is any physical trauma to the organ.
That's because the liver is one of the most sensitive organs in the body and can cause severe internal bleeding that may lead to death. However, in this case, we can see that this does not apply to our pig, as it appears to have a healthy liver.
Definitely, look, the liver has all its parts intact, doesn't have any visible physical trauma, and the weird color that you see is because of the preservatives that they put on these pigs. But for the rest, it looks very healthy. The next organ that we will be inspecting is the stomach. As you can see in this case, the stomach was cut near two areas. This is this incision we made it in the point where the stomach connects to the small intestines. And this other incision right here, you cannot see it as well because it is very tiny, but this is a connective tube that goes all the way up to the esophagus.
A further inspection of the stomach involves cutting it open to access its contents. This allows us to observe what was present inside the stomach just before death. The materials retrieved from the stomach can provide critical clues, especially in cases involving the ingestion of drugs or poisons.
But in this case, we will not be cutting the stomach open because we want to use this organ for later studies. So we will be skipping that part because we want to preserve the organ and give it as much of an use as possible. But as you can see from over here, from what I touched, not only is the stomach a sac that is containing liquid and acid inside, but I can definitely feel that there is stomach content in there, meaning that the pig had a meal before death. Okay, as we continue, let's take a look at the intestines. And yeah, I know you might be thinking it. This does not look much like human intestines, as in human intestines, the large intestine will be wrapped around the small intestine and closing the small intestine in the center, and your large intestine is also going to be shorter. In the pig case, the large intestine is going to be a little bit longer than in the case of the human. The other thing that we can notice is that the position of the organs is different, as I mentioned. And let me show you something. Maybe you guys got confused. I also got a little bit confused when I first took it out of the pig. I was like, wait, is this only the big intestine, or does this also have the large intestine? And look, I want to show you right here. This over here is the point of separation between the large and small intestine. This elongated section that we can see over here, you can see it is thicker. And it has a protective layer that is keeping it together. If I try to separate it, I can't, because there is an entire layer that is keeping all of that together. And that is because this part right here, that is the large intestine. And on the other hand, all of the rest of this mass, that will be the small intestine. And this is the connective part that connects the stomach to the small intestine. As you can see over here, this is where I made the cut, and that was the same thing that connects to the stomach as the previous cut that I showed you to the small intestine. And down here, this is where the large intestine continues to the rectum. This is a cut right before the rectum. And yeah, this is it.
A more in-depth inspection of the contents found in both the small and large intestines can give us insights into what the individual consumed orally within the last 24 to 72 hours. That's the average amount of time it takes for food to travel through the digestive tract. However, we will not be performing this inspection because we don't currently have the resources or the time to complete it. And let me tell you, detangling all of this is very time-consuming.
And now let's take a look at the brain. And I must tell you guys, be very careful when handling the brain because it is very soft and you don't want to break this first layer that is protecting the rest of the brain, because once this capsule is broken, the rest of the brain is just going to completely dissolve.
Analysis of the brain can determine detrimental clues that aid final information for the autopsy. For example, it can tell us if there was any brain disease, which provides a definite diagnosis of neurodegenerative diseases such as Alzheimer's, Parkinson's, or frontotemporal dementia. It also shows psychiatric disorders such as schizophrenia and major depression, and it shows physical changes in the brain that can be analyzed post-mortem. Lastly, it determines whether substances such as medications, drugs, or toxins affect the brain function.
That was a lot of information, I know, but don't worry, because that was not the case for our pig. After long studies on the brain, everything came back negative. This pig had a healthy brain structure, which indicated that there was a healthy brain function. Our last two organs to inspect are the kidneys. And bro, I feel like I have to say it one more time. These are bilateral organs, which means we have to
inspect them separately.
Good, I feel like you guys are understanding what's the deal right now. So let's start by taking a look at the left kidney. All right, gonna put it here on the table. And before we continue into looking at the parts of the kidney, let me bring in the right kidney. So let me show you something pretty cool right here. So in kidneys, you're going to have two connective tubes. The first one right here is which provides the blood that comes into the kidneys, and then it is filtered and the excess goes down the ureter, which is the same tube that is later down going to connect into one and come out of the urinary system of the pig. As we can see over here, the kidneys are just, we can only see these little beans, but they're actually layered organs, which means that what we see is just a capsule that it has inside the most important part of the organs, of these two organs, which are the medulla and the cortex. And they are the parts of the organ that do most of the job, pretty much.
By weighing and measuring the kidneys, we can rule out kidney failure, which would typically cause the kidneys to shrink and wrinkle. But as you can see, that's not the case here. These kidneys appear to be very healthy.
Well, that was a lot of information.
I know. But let's clarify something. The reason why we didn't do a more in-depth examination of these organs is because we wish to preserve them for future research.
That's right. These organs will be stored under the appropriate conditions to ensure that they can be studied for many more generations to come, reducing the amount of autopsies that we will have to perform in the future.
Which is also the core purpose of our videos. We don't want to just educate you guys, but we also want to reduce the number of sacrifices that are made for medical examinations or autopsies.
In this case, we were able to retrieve DNA sample out of tissue that we extracted from the body.
Additionally, we also sent a blood sample that we retrieved in order to send it to toxicology inspection.
Now it's just a matter of waiting for all of these results to come back to us, and that way we can look at a bigger picture of what happened.
But you guys know we don't make you wait. So let's discuss our findings right now.
Alright, here we got the files. These are all the results that came back from the lab, along with all the examinations and all the findings that we included in our autopsy. And let's wrap up what we saw here today.
There were no visible wounds, bruises, or marks found within the external examination of the body. In addition, the x-rays confirmed that no fractures were found.
Also, when the external examination started, all the organs were in their supposed position and no internal bleeding or organs were detected out of place.
The heart was healthy and none of its cavities or arteries were obstructed.
Well, the lungs were also intact. It says here, no water was found obstructing the bronchus. In addition to that, the vein vessels near the lungs, the necks, and the face region were not dilated nor exploded.
The liver also had a close to common weight, size, and color.
The stomach was not perforated. It did not contain any drug that might have been consumed after death.
The small and large intestines were in their regular shape and position and did not contain any drug that might have been ingested in the previous 72 hours.
Right, and both kidneys, as it says here, were in good standing. All their parts were closed to average size, and the weight too. Hey, that discards any kidney-related issues too.
Oh, so lastly, after doing several examinations of the brain, no irregularity was found pertaining to its structure.
Okay, but then what happened to this pig?
Wait, we forgot to mention the final and most important piece of information that we received from the toxicology.
What do you mean? What was that?
Samples collected from the specimen contain a high concentration of neuromuscular junction blockers, barbiturates, sodium channel blockers, potassium chloride, sodium hypochlorite, and formaldehyde.
Oh, wait. Those are all muscle relaxants, anesthetics, cardiac agents, and cytotoxic drugs.
That's correct. And that is the same mix of experience found in a euthanasia solution.
The mix of ingredients on an euthanasia solution. Wait, what does that mean then? That the pig was killed with an euthanasia injection?
Yeah, it was right in front of us all this time.
Wow. Now, let's change.
Your hair is eating. Is that how you say it?
Thank you,
Diva!
yes!
Diva!
Say it louder.
inspection of the external, internal parts of the body. Autopsies are used to discover the cause of death.